So then we'll move on to PacBio. Oh wait, of course. PacBio. So this is the PacBio RS. I kind of like this picture. It's a little bit sinister looking. You kind of feel that it might start talking to you and say stuff like, "I can't sequence that for you, Dave." The thing is actually, this screen here is actually about this height right here. So you can see that this is a very big machine. In comparison, the Lumina machines are about this big. Sorry for the people online. Basically just hit here. On the side here, this thing that's been circled in red, this is a black box. It is quite literally a black box in the sense that there is a lot of initial processing that I will talk about that goes on in this box, and PacBio really doesn't want you knowing what's going on in this box very much. This is the main part of the technology. This is the smart cell. You basically put a drop of DNA on top of this. Within that thing, there are 150,000 wells where the sequencing occurs. To get a little bit more detailed, this is sort of what happens in schematic mode. This is that smart cell that I was telling you about. Here are these things called zero-mode waveguides, which are the wells. I'll describe that in a little bit in the next slide. And what basically happens is you have a very heavily modified DNA polymerase that goes in here, your DNA template, and much like the Illumina system, but it's been done in a slightly different way. You have a bunch of nucleotides, and they have fluorophores attached to them. And basically, in a very similar way, lights are shot, camera image is taken, and then a lot of this stuff here that's going on happens in that little black box, and it basically does a lot of signal processing. Okay, so these zero-mode waveguides are these very sort of specially designed wells such that they want to have a single photon be able to enter in here and excite the fluorophore that's been released by the sequencing by synthesis to produce the signal that they're going to detect. The way this works is basically when you put in your reagents on top of the flow cell, it basically stochastically diffuses into the wells. Sometimes the well will be empty. Sometimes, and this is the case that we normally want, there will be exactly one strand of DNA with its polymerase. Sometimes there will be multiples, and this is a problem, and you can't really get useful data out of this. Over time, they've been developing little systems that have helped even out this distribution. So we've been getting better and better at having more of the wells actually be productivity 1. So what's actually going on inside of these wells? So the main part on the adapter side of the PacBio technology are these things they call the SMRTbell adapters. So here you have the fragment of DNA and its complement side, and what they have is these segments of DNA which have been looped. And these have a special primer region that will attract the DNA polymerase. So then it will start sequencing through here and across and over and down along, and it will continue doing this until the reaction ceases. Here is this process in a little bit more detail. Basically, instead of having flowed cycles like happened in the Illumina system, all of the nucleotides are present in the solution for the entire time. They're just in there, and the DNA polymerase does its usual thing, takes them and incorporates them, releases this fluorophore, and there's a signal that gets released, and it's detected. Now you'll notice here that these signals are not sort of the same in length. The PacBio system is very asynchronous. The Illumina system is very defined. Like I said, they do one base pair at a time. That cycle stops. They cap it or they uncap it, and they go to the next one. This thing is just continuously happening. It's a little bit more like it would be happening in nature. The polymerase captures the potential nucleotides and incorporates them as they go. Like I sort of alluded to earlier, this polymerase has been heavily modified. One of the many things, sort of the technologies that they sort of developed in order to bring the system was sitting around mutating the polymerase to give it various specific features. And throughout their various iterations of chemistry, they sometimes had the polymerase so that it is better at sticking with the DNA strand and not falling off. However, that version was a lot more susceptible to photo damage. So like I said, there's constantly this laser getting shot up into there, which tends to degrade the polymerase. It dies. In their most recent chemistry, they've tried to add this sort of thing that one of the people described to me as a cabbage, basically sitting on the side of the polymerase, and it's there to sort of absorb extra photons so that less damage occurs to the polymerase. Now this sort of diagram here of the signals is actually a bit of a cartoon, not really quite as complicated as it's actually sort of going on. So you can see here, because all of the fluorophores are present, there's always this background activity. And even this is not as bad as it kind of actually is. So what you're seeing here, you'll notice here that the yellow color, which will represent the A fluorophore, is actually on a very similar bandwidth as the C. Similarly, T and G are very close to each other in terms of their spectra. This happens to do with sort of limitations of finding fluorophores that are spread far enough apart in their spectra, and sort of certain cost limitations of the machine. Like I already said, the machine's pretty big. They sort of told me that it would be better if they had multiple lasers and some different optics. It would have made the machine even bigger and more expensive. So it's just one of these trade-off things. So what they do, they have a very sort of, they use a machine learning algorithm called the Viterbi algorithm, which is actually used a lot for things like cellular communications and stuff. And basically what they've done is they've taken a bunch of test sequences, internally, and trained all their machines and the algorithm to recognize certain patterns so that they can much more reliably figure out, hey, this is an A, this is a T, this is a G. Now one of their weaknesses, you can tell over here, see these two A's over here, how do they always know that that was one A and then rapidly another A? And that's one of the things that their algorithm has the most difficulty with. In this case, it managed to decide that this down peak here, I don't know if you can see it, was enough of a decrease in the fluorescence for a while to indicate it was actually two A's. What they tend to do is they bias themselves to guessing towards there being extra bases instead of missing a base. I think they feel that this is, it's better to go back and sort of figure out that you might have had too many A's than rather not have had an A there at all. I mean, an extra A to indicate that it was a homopolymer region. There's a lot more detail in that these are all the sort of different parameters they look at that they fed into their machine learning algorithm. So things like the duration, the intensities, the spacing from the other ones, the sets of spectra differences. So this is one of the things that is both good and bad about the PacBio system, is that they have a very specific error profile. They tend not to misread a base, right? The number of errors that they have that are just straight up, is a G, but it should have been an A, very low. Tends to represent maybe 10 to 20% of their errors. The majority of their errors are insertion errors, which is basically, and these tend to happen because there's a homopolymer repeat. So if there are multiple A's or multiple G's, they may get the exact number of those wrong, and they tend to guess over. You'll notice here in this deletion column, the multipath, I'll get in a little more detail what a multipath is. This is basically them comparing many reads of the same region and deciding, if there's been any sort of errors, which of those are actually the correct ones. There are two basic modes that it runs in.